Today we're going to be talking about the process of translation. Translation is protein synthesis. This is where we finally move from DNA and we get a protein that the cell can use. So this is what's encoded in the DNA. It's why the DNA exists. When we're looking at DNA and we're trying to read that code, translation is the final process, the final step in that process. It's literally reading our genes to give us the protein that our body needs, our cells need to carry out the important functions in day-to-day activity. So when we're going to go look at translation, first we have to make the assumption that transcription has already happened. Transcription is where we went from DNA to messenger RNA, right? We went through that process at the nucleus. We now have a messenger mRNA transcript, or mRNA transcript. That mRNA transcript leaves the nucleus and it's going to find a ribosome. The ribosomes are where we are going to build our proteins, and there's a lot of different things that are happening during this process. One new member or one new introduction we need to make for this process is this is where we see a couple of different parts of RNA or types of RNA that we haven't seen before. We've got messenger RNA, but we're also going to see here rRNA, which is ribosomal RNA, and then we're going to see tRNA, which stands for transfer RNA. And both of these play critical roles along with messenger RNA to get us to our proteins. So you really need all three types of RNA working together during this process. Okay, so messenger RNA is going to be a series of letters. It's going to be single-stranded. Okay, so it's going to look something like this. And along this strand, we're going to have different bases. Okay, so we're going to have A's, C's, U's, and G's. Okay, all the four different base pairs are going to be along this single strand of messenger RNA, which is complementary to the DNA. Came to the nucleus. These letters are going to be read three at a time. So for example, AGA, UAC. Each one of those three letters is referred to as a codon. And that codon codes for a specific amino acid. Each series of three letters or three base pairs codes for a particular amino acid. And there's a lot of overlap because there are 20 possible amino acids, but there are 64 possible codon combinations. So a lot of times, for example, AGA and AGC may both code for the amino acid valine. And this really works in our favor because sometimes if a mistake gets made and there's a mutation and a base pair switches places, we still wind up with the same amino acid. And if that's the case, then we can have a mistake in our genes and still wind up with the same protein. And so we're literally walking around with a mutation and we have no ill effects because of that. So anytime a cell is going to be undergoing translation, there's going to have to be a ready supply of amino acids there because we're going to be adding the amino acids to this growing chain as our messenger RNA is being built. We're actually going to build the protein piece by piece. So amino acids cannot recognize the codons on messenger RNA, and so that falls to the job of transfer RNA. Transfer RNA is actually going to carry the amino acid along with it. So a very simplified version of a tRNA molecule is going to look something like this. It's going to come down here and it's going to have three base pairs. And then on top, we are actually going to have an amino acid attached. It's just going to sit atop the transfer RNA molecule just like that. There will be a codon, or actually we'll call this an anticodon, that is complementary to the codon. So for example, if the codon on messenger RNA read AGA and this tRNA molecule were complementary, it would need to read U, which is complementary to A, C, which is complementary to G, and U. And so during this process, as we're going to see here in just a minute, that complementary base pair is going to match up with that, and then we'll be left with the amino acid. Okay, the amino acid is going to be left behind. So the tRNA, super important to this process. Without it, you're not going to get the amino acids, and without amino acids, you don't have proteins. Because remember, the amino acids are the building blocks of proteins. Amino acids are the monomers that build the protein polymer. So all of this is going to be happening on the ribosome. okay? On the ribosome is where all of this is going to happen. So let's leave our mRNA molecule there. What we're going to see happen is the mRNA molecule is going to arrive at a ribosome. And the ribosome looks something like this. And the ribosomal RNA makes up a majority of the ribosome, okay? This strand of messenger RNA is going to be fed through the ribosome. Messenger RNA is going to be fed through this ribosome. It's just being passed right through the ribosome. As it is fed through the ribosome, this is our first little codon right here, ACU. Okay, so let's move this into the ribosome. So that's inside the ribosome. In order for the process of translation to work, what's going to have to happen is while this is in the ribosome, the tRNA molecule with the complementary anticodon is going to come along. So that would be UGA. Okay, it's going to be attached to this tRNA molecule. And atop that tRNA molecule, it's going to have an amino acid. This tRNA molecule is going to come in while this is all happening very, very quickly. This is going to be slid in. The tRNA molecules are just going to be kind of floating around up here. It's going to temporarily dock. It's going to kind of acknowledge that they're complementary. And this amino acid is then going to be left. The tRNA molecule is going to go out of the ribosome, okay? And it will just be blank because this amino acid stays behind. These amino acids, as they stay behind, are going to start forming what we call a little polypeptide chain. And it's going to come out right out of the top of the ribosome. And every time there's a new amino acid, it's just going to get added. And this chain's going to grow longer and longer and longer. And you're just going to kind of see it feed out of the top of a ribosome. Okay? Kind of like a ticker tape. It's just going to continue to just add to this chain. It's just going to continue to print. Okay? And once we reach the end of this process, this chain of amino acids is going to be a protein. So how does it know where to start? How does it know where to stop? Okay, so let's kind of take a look at some of the finer points, and we can refer back to this diagram as we go into it. Translation is going to begin like transcription. The strand has extra base pairs that aren't part of the gene. So the first step there, which is initiation, assures that it begins in the right spot. The mRNA molecule is going to bind to a small ribosomal subunit right here on the ribosome. And there's going to be an initiator tRNA that binds to the start codon. And we're going to look for one particular codon in general on the messenger RNA, and that is AUG. AUG is the start codon. Okay? That is going to code for the amino acid methionine, and that is going to tell, okay, this is where the actual gene starts. Okay? You have to have an AUG. If you don't have an AUG codon, then you're not going to start this process. This is where things can get really tricky. If there's a mutation and it's supposed to be AUG, and the mutation changes this letter, that means you will not get the protein. Okay, so if there's ever a mutation right here, this could wind up being something severe because you've got to have this in order to start. So once we recognize this start codon, this causes the large subunit to bind and form a functional ribosome. So basically the two parts of this ribosome are going to connect right here over this messenger RNA once that code is read. The initiator tRNA finds one of the binding sites, which is called the P site, and that's going to hold the growing polypeptide. So that's going to hold these growing amino acids as they are being released by the new tRNAs which are coming in. So the tRNA is going to be coming in. The other binding site is what's referred to as the A site, and this is where the new tRNAs come in with the next amino acid. So we've got two sites there. Elongation is our next step. That's going to happen when the codon recognition of the anticodon from the incoming tRNA into the A site. So we recognize, okay, UGA is complementary to ACU. It's going to bond there. Then a peptide bond forms. And this peptide bond, this is why we call this a polypeptide chain. We're going to form peptide bonds in between these amino acids until we reach the end of this process. Once we reach the end, okay, then we're going to have, once we reach our stop codon, we've reached the end of translation. The process stops, and the ribosome is going to break apart, and this chain is now going to be released. Okay, it's released into the cytoplasm of the cell, and this now needs to be finalized. It's going to be finalized as a protein. It's going to need to be folded. It's going to need to be modified, so it's probably going to find a Golgi body within the cell to complete this process. Once the process is complete, we have our functional protein, and it can go out and do whatever job it is meant to do. So this is the process of translation where we're taking an mRNA transcript and we're making a protein within the ribosome of the cell. I'll see you next time.